This lab on parasitic worms and also arthropods that can act as vectors will be your last dry lab for this semester, and will be also your last logbook entry for this semester. So we should review the terms here, and then I'll show you some of the things that you need to look up for your to either fill out this sheet, or you can transfer everything, or this packet here, or you can transfer everything into your logbook entry. And after you're done with the logbook entry, so this one will be your last one, then you need to find out a way to digitize it, and I will make a little assignment box where you can then just upload your digitized version of your logbook. Okay, but so let's take a look at this lab here. So the dry lab. It will be covering, for the worms, it will be covering the phylum of the platyhelminths, the flatworms, and the two classes here in question are the class of the trematodes, the flukes, and the class of the cestoda, the tapeworms. And then we have another phylum, so the nematodes, those are roundworms. On the arthropod end, we have two classes that are being covered here. There'll be a class of the insects, the insecta, and then the class of the arachnida. Those are the spiders and the ticks. The way you always can distinguish insects from spiders is count the legs. Insects have 6 legs, and they include fleas, flies, mosquitoes, lice. Now the arachnids, they have 8 legs, so spiders have 8 legs, ticks also have 8 legs. Some of the terms, they were also covered in lecture, so maybe you want to look back to your notes on that. Hermaphroditic means that there's an individual, an animal in this case, that has both female and male gonads. So that would mean it's very common in worms that a worm would have both ovaries and testes, and of course that ensures high reproductive success. The term scolex, it means it's the head of a tapeworm. And the proglottids, those are the segments of a tapeworm. Now, two terms that you should definitely also know is intermediate versus definitive host. An intermediate host is one that harbors the larva or asexual stage of a parasite, versus the definitive host who harbors the adult or sexually mature form of a parasite. Now, larva, that is the sexually immature stage of a worm or an arthropod. And a vector, it's typically an arthropod that carries a disease-causing organism from one host to another, either via blood or sometimes a vector might carry a pathogen on their feet and then land on another surface and thereby transmit a pathogen. So the instructions for the lab are that each of the following worms and arthropods, you're supposed to examine slides or plastic mounts and complete the charts. Basically, the internet, again, is your best friend. You will find a lot of images there that are far better than what we actually have in the lab. So we're starting out here with Tinias saginata. This one is it's a beef tapeworm that you might end up getting if you're eating undercooked beef that was infected. There's a little story to tell about this one. Some people, they purposely infected themselves with tapeworms because they wanted to lose weight. You can certainly lose weight doing that, but it also bears obviously some risk and some side effects. So there's tons of information on the internet you find, including pictures, and you should make your own drawing. But the next one, the Clonorchis sinensis. This one is the Chinese liver fluke, and I think we had that one in a pathogen group as well. So you can find that one on the internet. I should make some drawings. Then here, Fasciola hepatica. This one here is a common liver fluke, mostly liver fluke. And then Schistosoma hematobium. That's a worm that causes schistosomiasis. I think there's a sketch at the end of this lab handout here. Let's take a look first at some roundworms. First one here is a giant one, is Ascaris lumbricoides, a giant roundworm. Then Necato americanus, that's a hookworm that you should look up. And Pterobius vermicularis. This one is a pinworm or threadworm. And then we have Trichinella spiralis. That one you may have heard about it. That is what the USDA is inspecting meat for. And so when it's having the label that it was inspected, mostly what they're looking for is Trichinella spiralis, because you can see the spiral arrangement there of this worm and in the meat if it's infected. And there's a picture in the back too. Then here is Wucheria bancrofti. This one causes lymphatic filariasis, usually has a mosquito vector. And then last one here, Toxocaria canis. That one is a worm that infects dogs mostly. You should just take a look up and see if you can find an image on the internet. Then so you have some information that you can read through, and let's take a look at the arthropod vectors that you're supposed to sketch. The first one is the body louse, not a pleasant partner. So to have, it transmits epidemic typhus. And then fleas, they are also transmitting quite a bunch of diseases, namely Yersinia pestis. That would be a flea. And you should try your best and make a drawing of a flea. How about ticks? Well, they transmit Lyme disease, as you know, and some other diseases as well. So here, the head and mouth part of a mosquito with the sucker. There we have the mosquito mouth part is right here. So you can draw that, or you can take this image. For the flea, you can sketch something like this here, or maybe make a copy and transfer it. The tick right here counts the legs. This one has 8 legs, so that will be an arachnid. And then here the louse that can transmit the typhus. So try your best to do the sketches. If you really can't get yourself to draw them yourself, then you can just use these samples that are given down here. Make sure that you can distinguish flea, tick, louse, mosquito, mouth parts, the long sucker. Here are some images that you might find useful for the parasitic worms. So here the scolex of a tapeworm, and then the segments, they're called proglottids. And here will be a Chinese liver fluke, the Clonorchus sinensis right here. So here, the scolex, the head part, and then the proglottids of a tapeworm. Then here you have a pinworm, the adults, where the female is larger than the male, and they kind of sort of live together, hug each other all the time. So there's a hookworm also over here. And then here some life cycle with some intermediate stages of a worm. And here's some more life cycles that you might find interesting. We had some of them in the lecture as well. And yeah, what I really wanted to show you was the encysted larva of the Trichinella spiralis. This is what they're looking for when they're inspecting meat. You're looking for these spiral kind of things that are occurring in infected meat. So here you have this encysted larva right here, and that is typical of the Trichinella spiralis, and meat gets inspected for the presence of these spiral kind of encysted larva. Alrighty, time to look up a bunch of stuff on the internet. You find way better picture than what I have here on this handout on the internet. Make your drawings, fill out the information. Again, this will be your very last logbook entry. Make it a good one, and also use this handout together with the or this packet here. Once you've filled it out, use it together with pathogen group number 6 to prepare for your next lab quiz, which will of course be online and will be next week. Since we have the lecture test, I will send you information how long this will be open and when you should be taking this particular quiz.